This lesson video is going to continue our exploration of cell structure and function. And in particular, we are going to look now at the permeability of the plasma membrane. The plasma membrane is selectively permeable, which means that it allows the passage of only certain molecules. And in this diagram, it's illustrating a number of ways that this can take place, and so we will look at each one of them in a little bit of detail. So molecules that can diffuse across the plasma membrane, they're shown with long back and forth arrows, and substances that cannot diffuse across the membrane are indicated by the curved arrows. So the molecules that can cross the membrane, then it's just showing them that some of them don't need any assistance in doing so. They can just simply diffuse across the membrane, whereas others cannot, and so they need some assistance in some way. So some substances, as I mentioned, they freely cross the membrane. So essentially, they move down the concentration gradient. So they move from high concentration to low concentration. At least that means that the substance itself. Okay, so we need to be clear in what it is that we're talking about, that substances, some can freely cross the membrane, moving from a location of high concentration for that particular molecule to lower concentration. So some substances are unable to freely cross and are transported by proteins or vesicles. So they may actually go up or against their concentration gradient. And we will look at some examples of that. So here's a chart then describing some of these here. And as you can see, some require energy, and this would be involved in active transport or exocytosis or endocytosis, which is exocytosis would be leaving the cell, and then endocytosis are materials that enter the cell. So the ones that don't require energy would be from osmosis, diffusion, or what we refer to as facilitated transport. So diffusion then is the movement of molecules down their concentration gradient. It does not require energy, and the rate of diffusion is affected by factors such as temperature, pressure, and molecule size. So a solution contains a solute and a solvent, and diffusion occurs until there's an equal distribution of solute and solvent. So you can see here where you have something that's concentrated in one small space within the solvent, and then over time, if left, then you will get an equal distribution of concentration throughout the solvent. Only a few types of molecules can diffuse across the plasma membrane, and so gases can diffuse across the bilayer. So oxygen freely enters the cells, and carbon dioxide leaves. In lungs, the oxygen moves from the alveoli to the blood into the capillaries. Osmosis is the diffusion of water molecules across a selectively permeable membrane due to a difference in concentration. So here you have a selectively permeable membrane, which allows some substances to cross, but not others. So in this case here, there's a net movement of water and changes in solute concentration on both sides of this particular membrane. So it's showing here that there's a lower concentration of water in the green area there, and so then over time you get a net movement of water from the bluish area into the greenish area, and this would be an example of osmosis, because there are some molecules in the green that cannot cross the membrane. So we have what we refer to as isotonic, hypotonic, and hypertonic solutions. And an isotonic solution, that has the same concentration of solute and solvent as the solution inside of the cell, which means that water will not enter or leave the cell. I mean, you will have some molecules moving back and forth, but for every one that goes out, one comes in. So the concentration then basically stays the same. With a hypotonic solution, you have a lower concentration of solute than solution inside the cell, which means then that water will enter the cell. With a hypertonic solution, that's where you have higher concentration of solute than solution inside the cell, and so therefore water will leave the cell. So hypotonic solution means that there's a lower concentration inside the cell. That means then that water molecules will enter the cell. With a hypertonic solution, that means that the solution outside of the cell has a higher concentration of solute, and so therefore you will have water leaving the cell in that situation. So here are some examples of that. We have isotonic, hypotonic, and hypertonic solutions here. And so with an isotonic solution, there's no net movement of water. With hypotonic, water will enter the cell, and with animal cells, if the concentration is high enough, then the cell will actually burst. So in a hypertonic solution, this situation where the solute concentration in the cell is lower than outside the cell. So that means then that you'll have a net loss of water, and so the cells will then shrink. In plants, this is important because it maintains what's referred to as the turgor pressure within the cell, which allows the plant to remain upright. So when a plant starts to wilt, the turgor pressure inside the cells have decreased, and so you need to put water back in. So transport by carrier proteins then, the plasma membrane, it stops the passage of most molecules into and out of the cell. However, biologically important molecules do need to pass, and they do so because of carrier proteins that exist in the plasma membrane. So carrier proteins are specific, and each binds to a specific molecule. Carrier proteins are required for both facilitated transport and active transport of substances across the plasma membrane. So this is facilitated transport, and so this is assists in transport of molecules across the membrane by binding to those molecules. This occurs down a concentration gradient again, and so therefore it does not require ATP. Active transport then, this is assists transport of substances across the membrane by actually binding to them, and so this occurs against the concentration gradient, and so therefore requires energy. And again, this would be from ATP. Proteins involved in active transport, they're often called pumps because they actually use energy to pump substances against their concentration gradient. And one of the very important carrier protein here is associated with the sodium-potassium pump. So it moves sodium ions to the outside of the cell and potassium ions to the inside of the cell. And what happens is you actually get a net charge from outside the cell to inside the cell. So there's a difference in voltage outside the cell as compared with inside the cell, and this is largely responsible, or it's the sodium-potassium pump that's largely responsible for that. So here you can see the workings of the sodium-potassium pump, and so the same carrier protein transports sodium ions to the outside of the cell and potassium ions to the inside of the cell because it undergoes an ATP-dependent change in shape. So 3 sodium ions are carried outward for every 2 potassium ions carried inward. Therefore, the inside of the cell is negatively charged compared to the outside of the cell. So here you can see, how that charge is set up by the sodium-potassium pump. Bulk transport, macromolecules are transported into and out of the cell by vesicle formation called membrane-assisted transport in energy-dependent processes. So again, energy is required for this. And exocytosis then is the way that substances will exit a cell, and endocytosis then is the method in which some of these macromolecules can enter a cell. So exocytosis then, what happens is a vesicle fuses with the membrane, and the substance it is carrying is secreted outside of the cell. And things such as neurotransmitters, hormones, and digestive enzymes are examples of substances that are secreted in this way. So you can see the vesicle basically just sort of combines with the membrane of the cell, and then the contents are then deposited outside of the cell. Endocytosis, on the other hand, is the way that macromolecules can enter the cell. So with endocytosis, cells take in substances by vesicle formation. So the plasma membrane folds in on itself and then pinches off to form an intracellular vesicle. And endocytosis can occur in one of 3 ways, and those are referred to as phagocytosis, pinocytosis, and receptor-mediated endocytosis. Phagocytosis is where the material being taken into the cell is large, such as a food particle or another cell, such as a bacterial cell that an immune cell is wanting to rid the body of. So this is common in unicellular organisms as well, and occurs in certain types of human white blood cells, as I mentioned. So it's often the white blood cells that take care of these foreign cells that happen to enter the body. And so one way that they take care of these is by phagocytosis. Pinocytosis, then, this is where vesicles form around liquid or very small particles, also common in blood cells and intestinal cells and plant root cells. So basically what happens is the cell membrane, it surrounds the particles and then pinches off, and then those particles then enter the cell in a kind of a vesicle. Receptor-mediated endocytosis is a type of pinocytosis, and it involves receptor proteins that only bind to certain molecules. So the receptors are in coated pits, and once vesicles form, they become uncoated and fuse then with the lysosomes within the cell. And empty vesicles fuse with the plasma membrane, and receptors return to their previous locations. So you can see the steps that occur in this kind of a process. So very, very similar as well to endocytosis. There's just further receptors that are involved with this particular process. So that will end our look at cell structure and function. And so we've looked at the plasma membrane. We've looked at many of the organelles within the cells, and the cytoskeleton as well. And so this is a summary of the things that we have covered in this particular lesson. And we will continue to look at things involving cells in the next section.